Welcome to MycoTalks. I'm Joe Heitman from Duke University, and it's really a pleasure to be here today with my co-host, Gordon Brown. MycoTalks is a broad seminar series that focuses on diversity. There are two 30-minute talks at each of the MycoTalks, and we ask that you submit your questions through the Q&A function that the moderators will be able to see and identify which speaker your question is directed to. And I'll just note that recordings are available at the MRC YouTube channel for others to view. I want to acknowledge the organizing committee that's been central in putting together the series, and also the University of Exeter MRC Center for their leadership here. We have two speakers today. This will be Agostinho Carvalho and Duncan Wilson. I want to also introduce that the next MycoTalks series will be held on July 27th. Darius Armstrong James and Aaron Mitchell will be the two speakers, and you should register for this ahead of time. I'll also note that there will be no MycoTalks held in August. We'll have a 1-month break, and then season 4 will start up again on September 28th. And for the trainees, there's the Medical Mycology Trainee Seminar Series. The organizing committee is featured here, and please reach out to them if you're interested in giving a talk for the series. So it's a great pleasure to be able to introduce our seminar speaker for today. This is Agostinho Carvalho, who's one of our two speakers for MycoTalks. And Agostinho is a principal investigator at the Life and Health Sciences Research Institute of the University of Minho in Braga, Portugal. And his team is studying the effects of inter-individual genetic variation on molecular and cellular processes of antifungal immunity, and harnessing this information to develop personalized approaches for the management of fungal diseases. Furthermore, they're also analyzing the genetic regulation of immunometabolism related to fungal infections, and they've also played an important role in understanding fungal co-infections in the context of COVID. Welcome, Agostinho. We're very much looking forward to your talk.
Thank you very much, Joe. I'm just going to share my screen. So I hope that you are seeing it okay.
Looks terrific.
Okay, so today I'm going to talk a little bit about some work that we've been doing over the past few years about some metabolic circuits that we identified to be very important in the regulation of immune function in response to aspergillosis. Now, I don't think I need to stress the importance of aspergillosis to the audience of MycoTalks. We focus mainly our studies on infections caused by aspergillus. And as you may know, Aspergillus fumigatus has been included in the critical group of the recent WHO priority list. This has many reasons for it. Of course, we are mostly interested in the infection that affects immunocompromised individuals, leading to very important mortality rates. But of course, we can also not forget about the impact that these infections cause also in other types of patients with chronic and allergic forms of aspergillosis. Susceptibility to Aspergillus infections has been historically thought to rely on the combination of different factors, both from the environmental perspective, also variables related to the pathogen itself, including a number of virulence traits, ability to acquire drug resistance, which is something that we are now realizing also to be very, very important, but also a number of variables that exist in the host and that somehow also determine the risk of infection. As Joe mentioned in the beginning, our group has been mostly interested in the genetic regulation of susceptibility. But more recently, we have also focused a number of our projects on understanding the role of metabolism, particularly of immune cells, and how this regulates the immune response to this infection. Now, if you think about metabolic activation of immune cells, we immediately think about the Warburg effect, or what other people also call aerobic glycolysis. So this is a process that is very well known from the cancer field, and in which we have basically a cell in a resting state, a macrophage or a monocyte, that relies mostly on oxidative phosphorylation to generate energy. And in circumstances of infection or stimulation, these cells rapidly shift their metabolism towards glycolysis. So this is essentially a very fast way of acquiring energy to basically answer to the infection. It is not as efficient as OXPHOS, but it allows a rapid acquisition of energy required for the activation of specific antifungal effector functions. Now, it is not surprising that most of the knowledge available to date has been uncovered with regards to infections caused by Candida, specifically Candida albicans. And what is most interesting, at least in my perspective, is that many of these studies have actually demonstrated a very important role for glucose metabolism across different cell types, ranging from monocytes to natural killer cells to neutrophils, and also more recently, also epithelial cells. Now, another interesting aspect of the metabolic features that occur during fungal infection is that some of these pathogens, particularly Candida albicans and also more recently shown for Candida auris, is that they somehow also evolved in a way to basically counter these mechanisms deployed by the immune cells. Essentially, Candida are able to outcompete immune cells for glucose, depleting this essential nutrients and triggering macrophage cell death, therefore contributing to their persistence and progression of infection. So based on this, one of the questions we had in the lab a few years ago was whether immunity to aspergillus could also be regulated at a metabolic level. And we took advantage of a model that we have in the lab, so using human macrophages infected with Aspergillus fumigatus spores, and we did some transcriptome analysis of these cells. And basically, to make a long story short, we found that there were a number of genes involved in glucose metabolism that were upregulated during infection. Here shown, for example, the glucose transporter, hexokinase, and also PFK-FB3, a very important regulator of glucose metabolism. Now, these changes occurring during infection were not only observed at the transcriptional level. We also could detect these changes if we quantified both glucose consumption from the culture media and lactate secretion into the culture media, as you can see here. To further confirm that this was indeed the activation of glycolysis, we also quantified a number of glycolytic intermediates. And essentially, we saw that the infection was also leading to an increase in these metabolic intermediates. So clearly, this seems to show that infection with Aspergillus fumigatus is reprogramming macrophage metabolism. Now, one way to confirm whether this is important for antifungal immunity and effector functions is basically to disrupt the activation of this process. And we did that, as many groups do, by using this analog of glucose, 2-DG, 2-deoxy-D-glucose, which basically inhibits hexokinase 2. And we found that by using this during the infection, we can decrease levels of lactate being secreted, and this nicely correlates also with an impaired ability of these macrophages to get rid of the fungus, to kill the fungus. Now, this is also true if we measure cytokines being released during infection with an overall decrease of a number of pro-inflammatory cytokines. This is also true not only in vitro, but also in vivo. If we use a similar strategy by treating mice with this compound before infection, we can notice that 24 hours after infection, we have an increased fungal burden in the lungs, and this is also correlated with an impaired secretion of cytokines in lung homogenates, similar to what we have observed in vitro. So clearly this seems to indicate that glycolysis is required for this protection against Aspergillus fumigatus infection. Now, what about the mechanisms that underlie this process? So for most of what we know in terms of Candida infection is mostly due to the activation of Dectin-1 with beta-glucan. This has been mostly shown for processes related to trained immunity or innate immune memory. And we were wondering whether there were similar mechanisms also in place during Aspergillus infection. So we started by just, repeating the same experimental model, but this time not only using spores, but using different fungal morphotypes. And actually, we came to a very interesting result, which was the fact that indeed lactate secretion and therefore glycolysis activation was observed when we used dormant spores. And this phenotype kind of was being inhibited after germination. So suggesting that there was some kind of component in the dormant spores that was responsible for glycolysis activation that was somehow lost during germination. Now, to identify the compound that could be responsible for this phenotype, we took advantage of two different cell wall mutants. So a ROD1 mutant without the external ROD1 layer, and the PKSB mutant, which is an albino mutant that lacks melanin. Doing the same infection model, we found that only in the absence of melanin, we could see that lactate was not induced. And strikingly, by just coating ROD1, PKSB double mutant spores with melanin, we would be able to rescue lactate secretion in a dose-dependent manner. Now, this not only rescued the activation of glycolysis, but it also rescued the expression of glycolytic genes and also cytokine secretion in response to infection. So this seems to suggest that fungal melanin is indeed required for the metabolic responses to Aspergillus fumigatus. Now, at the time, Gordon was working on the identification of a C-type lectin receptor as the receptor for DHN melanin of melanized fungi, including Aspergillus. And importantly, a loss of function genetic variant in this human receptor was found to increase the risk of certain patients to pulmonary aspergillosis. Now, this was confirmed as a loss of function variant because by taking monocyte-derived macrophages from healthy donors and infected them with spores, we noticed that the presence of the variant led to an overall defect in cytokine production in response to infection. However, surprisingly, we did not find any effect in terms of lactate secretion and glycolysis activation. So apparently, recognition of melanin by MELAC is largely dispensable for the phenotype we were observing. So we then next moved to try to understand what were the signals responsible for this metabolic activation. A number of pathways have been proposed before for Candida albicans, including AKT, mTOR, and IF1 alpha. And it was not surprising to see that in the case of melanin-mediated activation of glycolysis in the case of Aspergillus infection also relied on the same players. You can see here in the red boxes that the PKSB infection actually led to lower levels of S6 kinase, which is a downstream target of mTOR activation, but also phospho-AKT were markedly decreased during infection with the albino mutant. And also true was the fact that the infection with the albino mutant also did not trigger nuclear translocation of IF1 alpha, as well as did the infection with the parental Q80 strain. Now, interestingly, going back and using the same coating model, using the ROD1 PKSB mutant coated with melanin, we found that it was possible to actually rescue mTOR activation and also IF1 alpha translocation. I'm not going to extensively show you all the data, but basically if we block any of these molecular regulators, we lose basically this glucose metabolism activation. So suggesting that there's the activation of this immunometabolic signaling axis, and this is required for the immune response to aspergillus infection. Now, what does how can we link the activation of these metabolic pathways with the presence of melanin? So work by George Hamilos had previously shown that melanin is a very important molecule within the phagosome, as it activates a number of processes, including the sequestration of a number of ions, such as calcium. And George showed that this was very important for activation of LC3-associated phagocytosis. So we started by confirming that indeed, in the absence of melanin, we would see scattered calcium ions across the cell, whereas whenever there was melanin present, these calcium ions were mostly sequestered within specific organelles or phagosomes. And actually, by using a number of pharmacological inhibitors, for instance, EGTA, to block extracellular calcium, we found that this was not very important for the phenotype we were measuring. But when we indeed modulated intracellular calcium and intracellular calcium signaling by using tapsigargin to basically deplete endoplasmic reticulum calcium, or W7 to block calmodulin signal, we can see that it is possible to rescue the defect presented by the infection with the PKSP spores in terms of lactate secretion. So apparently, sequestering of calcium is required for the activation of macrophage glycolysis. Now, the next step was to try and find a link between the ability of melanin to sequester calcium and the activation of metabolic pathways. So we started by just quantifying the levels of mTOR in these cells and how calcium inhibition or inhibition of calcium signals could impact its levels. We found that indeed there was a very marginal difference between the Q80 infection and the PKSP infection. This was somehow restored by calmodulin inhibition. However, the most interesting results that we obtained was when we quantified the number of phagosomes that were actually coated with mTOR during infection. And you can see that whereas infection with PKSP leads to a defect in the number of these mTOR positive phagosomes, if we block calmodulin signal, we can nicely rescue these numbers, suggesting that these two processes are interconnected. And actually, by modulating calcium signals, we can also rescue the secretion of cytokines in response to the PKSP infection. So apparently, the recruitment of mTOR to the phagosome is required to basically allow calcium signals to regulate the reprogramming of macrophage metabolism. So basically, with this work, we identified a mechanism in which melanin of Aspergillus fumigatus somehow activates, it could activate a pattern recognition receptor that we still do not know. But inside the phagosome during germination, we think that the phagosomal removal of this melanin sequesters calcium ions, and this effect will then lead to the recruitment of mTOR that will then regulate a number of changes that will lead to the metabolic reprogramming of the cell, leading to lactate secretion, but at the same time regulating a number of effector functions, including production of pro-inflammatory cytokines. Now, this was very interesting because we could demonstrate the importance of this process both in vitro and using animal models. But one of the questions that we also had was whether this process was important also in humans, in human patients. And one way to basically understand this in patients is to somehow look at genetic variability of these patients and how this influences the ability of the cells to reprogram their metabolism. So we had identified in this work a number of candidate genes that we found to be very important in the immune response or in the immunometabolic response to Aspergillus infection. So what we did was to team up with Mihai Netea and basically run a functional genomics analysis of cytokine production in response to aspergillus infection. So these are PBMCs that were infected with aspergillus, and essentially what we did was to search for genetic variants across these metabolic candidates and identify variants in these genes that could modulate cytokine production. And it was very interesting to notice that from all of these candidates that there was a marked enrichment in genetic variants in this gene PFK-FP3 that I mentioned before. It's a very important regulator of glucose metabolism. So here shown just a couple of examples, and you can see that there are interesting genotype dependent effects on cytokine production according to these variants in this metabolic gene. Now, we then proceeded by using a cohort that we have available in the lab, the IFIGEN cohort, which is basically a cohort of stem cell transplant recipients at risk of developing invasive pulmonary aspergillosis. And basically we found that the donor genome, the presence of one of these example variants that I mentioned before, namely the TT genotype here noticed in the red line, actually increases the risk of infection after transplantation. And this is just also to show you that after multivariate analysis and correction for a number of clinical factors, actually the presence of this donor TT genotype as a comparable risk increase comparable to acute graft-versus-host disease, which is a very important risk factor in these patients. From a mechanistic point of view, what we did next was to take, again, human monocyte-derived macrophages from healthy donors, screen them for the presence of this genetic variant, and basically evaluate the expression of PFK-FP3. Now, it was interesting to notice that even in unstimulated cells, we could already see a defective expression of the protein. This was also true when we performed the infection. And this correlated very nicely also with the levels of secreted lactates. So showing also that unstimulated cells just during the 24 hours of the infection protocol, but in this case without any infectious stimulus, also produced lower levels of lactate. And this was maintained during infection. This phenotype, this metabolic phenotype, was translated into a mild effect on the candidiacidal activity, but most of the effects that we saw in terms of antifungal effector functions were actually at the level of cytokine production. I'm not going to show you the whole the in vitro data, but just to show you the relevance of this process in patients with invasive pulmonary aspergillosis. Here you can see the levels of a number of very important cytokines that we detected in the BAL samples from these patients. And for instance, for IL-1 beta, IL-6, IL-17, and IL-22, the TT genotype in red, which was the one that increased the risk of infection, is also the one that is associated with lower levels of these cytokines in the alveolar fluid. So apparently PFK-FP3 is thus a major regulatory node of immunometabolic responses to human aspergillosis. Now, if we think about metabolism and the complexity of metabolism, it's very reductive to think that glycolysis and glucose metabolism is the only important aspect of what is going on in an immune cell during an infection. And it was with this in mind that we decided also to look at different pathways. So I'm just going to show you a couple of examples. We are working on others, but basically a recent project from the lab we took a look at, again, our macrophage transcriptome, and we found that in terms of glutamine metabolism, we selected a number of genes involved in glutamine metabolism, and we found that infection actually led to the emergence of a number of differentially expressed genes. Things became even more interesting when we quantified the levels of glutamine during infection, and we noticed that actually glutamine was rapidly consumed in the culture media during infection, something that did not occur when cells were left uninfected. Now, as most of you know, glutamine is a very important amino acid because it can feed the TCA cycle through alpha ketoglutarate, and in this case, to allow the cell to use glutamine as an energy source. And basically, what we did next was to quantify a number of these metabolites from the TCA cycle, and you can see that a number of them, including alpha ketoglutarate, succinate, and fumarate, were also increased during infection with Aspergillus fumigatus. Now, this was very interesting to see, but we then manipulated glutamine metabolism to show whether this was important for antifungal immunity. You can see here in the dark blue bars, this is the normal amount of glutamine that we use in the culture media. We then did some experiments by supplementing with glutamine or by eliminating completely glutamine from the culture media. And you can see that cytokine secretion clearly depends on glutamine, but it is not favored if we actually supplement the culture media with it. And the same happens to specifically to phagocytosis. No significant effect was found regarding candidiasis activity. Now, I'm not going to give you all the details, but we did similar experiments by using the pharmacological inhibition of glutamine metabolism at different steps, including, the transporter of glutamine and also glutamine synthase, which is the first enzyme that converts glutamine to glutamate. Now, we then next moved to some in vivo analysis of glutamine metabolism and did a similar experiment. We treated mice with BPTS, which is a glutaminase inhibitor. And you can see that whereas inhibiting glutaminase leads to an increase in the fungal burden in the lungs of these mice, glutamine supplementation does not seem to do anything really relevant. This is just to show an example of the histology of the lungs of these animals. And likewise, we do see a similar effect in terms of cytokine production with lower levels of cytokines whenever we inhibit glutaminase, whereas glutamine supplementation does not really lead to an improvement in the cytokine secretion. This is also just to show that there are a number of genetic variants across glutamine metabolism genes that are also cytokine QTL, so basically genetic variants in glutamine metabolism that influence cytokine production in response to Aspergillus, although at the moment we don't have any evidence that these variants are important in the actual development or in the actual risk of developing aspergillosis in patients. Now, for the last couple of minutes of my talk, I'm going to just show you very briefly a glimpse of a project that we have been developing in the lab, which is basically to understand whether known mechanisms of defective antifungal immunity can also have a metabolic basis. And you probably know this. We for many years we've been studying the role of an important molecule of innate immunity, which is the long pentraxin 3. So this is a component of humoral innate immunity, and it is most well known by having a role in pathogen opsonization and regulation of inflammation. And in the case of Aspergillus, is that it has been shown to have a non-redundant role in mouse models. So mice die from infection with Aspergillus, even if left immunocompetent. And also genetic variants, importantly, loss of function genetic variants are also important risk factors for infection in immunocompromised patients. Now, interestingly, if we take cells with these variants and we put them in vitro, this is data regarding neutrophils, you can see that giving its role as an opsonine, we do see a defect in Canidia internalization. But what is most interesting, at least for us, was that also the killing activity was also defective, even when these data and these results were corrected for the defect in Canidia ingestion. So could this defect be due to some metabolic impairments in these cells? So we first started by actually confirming that monocyte-derived macrophages, bone marrow-derived macrophages from pentraxin 3 knockout mice, they had a defect in phagocytosis, but also the same defect in the killing activity we noticed before in neutrophils. By basically performing the infection and in this case by opsonizing Canidia with recombinant pentraxin-3. So again, restoring the levels of pentraxin-3 to normal, we can rescue both of these phenotypes. So the question was, is this due to changes in metabolism? And actually, we found that pentraxin-3 bone marrow derived macrophages actually secreted lower levels of lactate, so apparently impaired glycolysis. This was again rescued by infecting the cells with opsonized spores. Similar findings were obtained for the ADP-ATP ratio, which is also another measure for glycolysis activation. But strikingly, we did not see a relation between lactate secretion and glucose consumption, as there were no significant changes here. So this means that glucose is actually being used by the macrophage, but it is not really feeding glycolysis in the same way in wild type and knockout cells. So to further look into this, because this was suggesting that other pathways were being activated, we developed this targeted metabolomics approach in the lab that allows the quantification of approximately 60 different metabolites. And we found that whereas in the wild type, as expected, we have an increase in metabolites and glycolytic intermediates. In the case of the knockout cells, there was instead a skew towards the intermediates involved in the pentose phosphate pathway. So it does appear that not only we have a defect in glycolysis, but we do have a gain of function of the pentose phosphate pathway in cells that are deficient in the long pentraxin-3. And to further confirm this, we actually measured the gene expression and the activity of the rate-limiting enzyme of the pentose phosphate pathway, so G6PD, glucose 6-phosphate dehydrogenase. And again, we found that infection of the pentraxin-3 knockout cells would lead to an increase in the pentose phosphate pathway, and this was somehow suppressed when we used pentraxin-3 opsonized spores. To further confirm this, we used a inhibitor of G6PD, 6AN, and we restored levels of lactate secretion to normal. And this was also in line with the rescue of the candidiasis activity defect, and also seemed to rescue a number of cytokines, most notably IL-1 beta. So we're now still working on the molecular processes that are basically responsible for a role of pentraxin-3 in regulating this metabolic skewing. But I would like just to finish with a summary for these projects that I've discussed with you. So it is apparent from the results that I showed that there are different and likely interacting metabolic pathways that are required for antifungal immunity. We know that recognition of fungal ligands, this has been shown for beta-glucan in the context of trained immunity, and melanin in the context of aspergillosis is important in the regulation of these processes. We know that genetic variants in metabolic genes are important for the risk of infection, and this occurs because this metabolic homeostasis is disrupted. And I showed you just a glimpse of an ongoing project that seems to demonstrate that molecules involved in the opsonization of spores also exerts a role in antifungal activity by regulating metabolic mechanisms. And I hope to at least somehow convince you that immune cell metabolism may represent an interesting opportunity for host-directed medical interventions, not only from the therapeutic perspective, but also from the risk stratification perspective. And with that, I'd like to finish by thanking the members in the lab, particularly Rita, Samuel, and Daniela, which have been responsible for the projects that I brought to you today. Several collaborators, both at ICVS, the IFIGEN study group, which is a very important cohort for us, and I would like to publicly thank the clinicians that have been critical for this, and also a number of international collaborators for their support over the years, of course, funding agencies, and you for listening.
Fantastic, Agostinho. Thank you so much for this scintillating talk. And questions are already coming in through the Q&A. I'm going to hand it over now to my co-chair, Gordon Brown, who will introduce our second speaker.
Thanks very much, Joe. And just to remind all of you, please, when you submit your questions in the QA, just to put the speaker name down at the front, just so Joe and I at the end can go through them and be able to identify which question is for who. So it's with great pleasure that I'm able to introduce my colleague here at the MRC Centre at the University of Exeter, Duncan Wilson. Duncan is a Wellcome Trust Senior Research Fellow here in Exeter. And Duncan is particularly interested in nutritional immunity and the fight between nutrients between the host and the pathogen. And he's played a really important leading role in identifying the first Candida albicans Zinc system and how the Candida is able to scavenge this essential micronutrient from the host. And today he's going to tell us about a brand new exciting news story, which he's pioneered, which gives us some new insights into pathogenesis of PVC. Over to you, Duncan. You're muted, Duncan.
Yeah. Okay. Thanks very much for the introduction, Gordon. It's just okay. Laser pointer. There we go. Okay, so yes, the title of my talk is that zinc prevents vaginal candidiasis by inhibiting the expression of an inflammatory fungal protein, or more specifically, Candida albicans PRA1. Zinc-4 drives vaginal inflammation. So this is a graphical representation of my talk. I'm going to show you that a low zinc environment triggers the expression of PRA1, which produces the secreted zinc binding protein, the Zinc-4, and that the PRA1 Zinc-4 drives vaginal inflammation. And just point out that more or less all of the experiments I'm describing today were done by Elena. Okay, so first of all, these 3 points. Why is a low zinc environment relevant during infection? So Gordon touched on it. It's because of nutritional immunity. So just like us, the microbes that colonize us and potentially infect us have to get zinc in their diet, right? You can't make it, you need to eat it. We eat it in our diet with our food. Pathogens obviously have to get it from the host tissue. So what us mammals have evolved to do is restrict or try to restrict microbial access to key micronutrients such as zinc, and that process is called nutritional immunity. And of course, pathogenic microorganisms must have evolved countermeasures to overcome nutritional immunity, get zinc from the host, and grow and cause disease. So I've been interested in this interplay in human pathogenic fungi, in particular Candida albicans. Okay, second point, what is PRA1? PRA1 is a gene which encodes a protein, and the protein is secreted from the cell and scavenges zinc from the local environment of the host. So it delivers zinc to the fungal cell so that the fungal cell can grow. And we've characterized the system in Candida albicans, but I'll get to it at the end. It's also present in several other fungi as well. And very importantly, in the context of this talk, before we even started working with PRA1, there was this nice paper showing that the PRA1 molecule can be recognized and targeted by human neutrophils. Okay, finally, why are we interested in vaginal inflammation? Well, vaginal candidiasis, although not fatal, is probably one of the most common serious, I would argue, the most common serious fungal infection on the planet. There's an estimated 500 million infections per year, so it's a really staggering number. And the symptoms are driven by inflammation and are associated with a non-protective neutrophil response. So unlike in many other manifestations of fungal infection, the neutrophils coming in to the site of infection during vaginal candidiasis are non-protective. And that's kind of like stemming from the work of Paul Fidel, Jack Sobo, and David Denning, some of these statements I'm making here. So my idea here, if we can prevent inflammation, can we prevent disease? Right, the initial model for this was basically stitching together a few bits of disparate information. So we knew that PRA1 could be recognized by neutrophils, and we know that neutrophils contain very high levels of calprotectin. Now, calprotectin is a host protein which can bind zinc and attempt to restrict its access to microbes. So calprotectin is one of the major arms of zinc nutritional immunity. And we know that calprotectin-mediated zinc restriction drives PRA1 upregulation. And, you know, we start at the beginning again, PRA1 recruits neutrophils, more calprotectin, more zinc restriction, more PRA1. So you can see how this could quite rapidly form a pro-inflammatory circuit, right? Kind of closed loop of inflammation. So that was the starting hypothesis. But first of all, we had to address a very first simple but important aspect to the story, and that was PRA1 expression. Because quite a long time ago, I was still in secondary school back in 1998, when Candida albicans PRA1 gene was first named, it was named pH-regulated antigen, and that's because this study showed that PRA1 gene is upregulated at neutral alkaline pH and not at acidic. And that could have been an issue for our hypothesis, because the human vagina is unique amongst mammals. The human vagina is acidic, around 3.5 to 5, whereas almost all other mammals are neutral alkaline, and certainly mice and rats that we would be doing experiments in have got a neutral vagina, and of course all of our tissue culture media, RPMI, DMAM, etc., is also neutral. So we could have found some pretty cool stuff in mice, rats, and tissue culture infection experiments that would be completely irrelevant to human disease. So first we had to show that Candida albicans PRA1 could be expressed at acidic pH. So here we infected A431 vaginal epithelial cells in standard DMAM media. That's in pH 7. PRA1 gene is strongly upregulated. And what we did, surprisingly, this hadn't been done before, because the human vagina is well known to be acidic. We did infection experiments at acidic pH, at pH 5. And just like at pH 7, PRA1 expression is very strongly induced during vaginal infection, even at acidic pH. And at both pH values, we were able to switch off expression of the gene by supplementing the media with some zinc. So at least during this tissue culture infection model, PRA1 expression is predominantly zinc regulated, more so than pH. That, of course, was important so that we were able to basically model human disease in the lab. Okay, so the next question was, is PRA1 expressed in the clinic in women? So together with our collaborators, we collected vaginal swabs and of women infected and colonized with Candida albicans, and we measured a number of host factors, pro-inflammatory cytokines, etc., number of fungal factors, PRA1, of course, and other let's say better known virulence factors like EC1, HWP1, etc. Obviously, we don't have time to go into all of the detail of this matrix and all of the correlations. So I'd just like to highlight obviously what I find the most important, and that was PRA1. So we found that PRA1 was indeed strongly expressed in a subset of women. So in 7 out of the 17 women, PRA1 was upregulated compared to in vitro conditions. Not only was it upregulated, but its expression was determined by QRT-PCR, positively correlated with a key inflammatory cytokine IL-1 beta. Not quite significant, but a positive correlation. And then a very strong and significant correlation between PRA1 expression and IL-8, which is the key neutrophil chemoattractant cytokine. So PRA1 is expressed in women and positively correlates with inflammatory markers. And for all you EC1 candidiasis aficionados out there, I know there's many of you, we did actually see EC1 expression in these samples in a subset of women, around 4 or 5. However, expression of EC1 did not correlate with the inflammatory cytokines in these individuals. Okay, so PRA1 is expressed by Candida albicans during vaginal infections in humans. So the next question was, is PRA1 driving vaginal inflammation? To do this, we set up a standard transwell-like chemotaxis system where we culture Candida albicans in a media where we know it expresses PRA1. We collect the supernatant, filter sterilize it, put it into the lower compartment of the chemotaxis plate, put labeled neutrophils in the top, and the idea is if we have a chemoattractant in the bottom, the neutrophils come through in response to it, and we're able to assess neutrophil chemotaxis by measuring fluorescence in the lower compartment. Okay, so the first part of the assay, the generation of the fungal supernatant. So we cultured Candida albicans for 5 days in RPMI media, and we did that with Candida albicans wild type strain, which of course is 2 copies of PRA1, a PRA1 deletion mutant where I've deleted the gene, and a PRA1 revertant where I've put the gene back. All 3 strains grow perfectly well. PRA1 is strongly expressed in the wild type, obviously absent in the mutant because I've deleted the gene, and then PRA1 comes back in the revertant. We also see a similar overall secretome amongst the 3 strains, but of course only wild type and revertant secrete PRA1. Okay, next part of the assay, we take that supernatant and we put it into the transferal-like system. So fluorescence indicates neutrophils coming through towards our chemoattractant. We have the negative control, no neutrophils come through. We have a positive control, IL-8, neutrophils come through. We have PRA1 containing wild-type supernatant, neutrophils come through in response to that, but we delete PRA1 and no neutrophilic response. And when we put PRA1 back into the mutant, we get neutrophils again. So PRA1 is essential for neutrophil infiltration in this ex vivo model. What about in vivo? So to answer this, we return to the mouse model. So we vaginally colonize or infect mice with Candida albicans wild type and with PRA1 deletion mutants, and then we wait and we measure several parameters. So we see that both Candida albicans wild type and the PRA1 gene deletion mutant are able to colonize the mouse vagina to comparable levels. But whilst Candida albicans wild type infection causes robust upregulation of IL-1 beta and CXCL2, that is the murine ortholog of human IL-8, both of those come up strong in response to Candida infection. Lots of neutrophils come in to the vagina of Candida albicans infected mice. Whereas when we delete PRA1, despite fungal colonization being more or less the same, no IL-1 beta comes through, is expressed, no CXCL2 is expressed, and no neutrophils come through. So deletion of PRA1 abrogates the molecular and cellular basis of inflammation in the mouse. So PRA1 is essential for inflammation in our tissue culture model, and in vivo, can we block it? Okay, I'd just like to emphasize, so I'm a molecular microbiologist, so I'm not an immunologist, and I'd just like to emphasize how important it is to understand the function of your gene and protein of interest, because it's only because we understood the function of PRA1, i.e. its zinc scavenging properties, that we understood how to regulate it. And of course, the zinc scavenger is under very tight transcriptional regulation by its substrate. So we know that we can switch off PRA1 with zinc. Okay, so that's what we did. We cultured Candida albicans again in the RPMI media, which we're about to use for our chemotaxis assay again. And we see that PRA1 expression goes up strongly in this RPMI media. But if we pop a little bit of zinc into the media, the Candida albicans downregulates PRA1 to 0. And then we take the supernatant from Candida albicans wild type, and Candida albicans wild type treated with zinc. We do our chemotaxis assay again. Again, we get a strong neutrophilic response towards Candida albicans wild type supernatant. We add some zinc, no PRA1 expression, no neutrophils come through. So we're able to prevent neutrophil influx in this model by adding some zinc. Okay, so PRA1 is essential for inflammation. Can we block it? Yes, we can. Can we block it in vivo? So we do our mouse experiment again. We infect mice with Candida albicans wild type only this time, but we do it in the absence and in the presence of some zinc supplementation. And we see that this is actually the first time we've measured this. Candida albicans strongly upregulates the PRA1 gene upon vaginal infection, but we add some zinc, the PRA1 gene goes down significantly. Again, we see high levels of IL-1 beta, CXCL2, and lots of neutrophils coming into the Candida albicans infected vagina. We treat with some zinc. Both pro-inflammatory cytokines expression goes down, and neutrophils are no longer recruited at high numbers. So turning off PRA1 with some zinc blocks inflammation as well. What about in the clinic? So that was in ex vivo models and in vivo mouse models. What about in humans? So we, or rather our collaborators, did this study. So there is a zinc vaginal moisturizing gel called Juvia or Uvia, right? And it contains about the same amount of zinc in it as we were using for an infection experiment. So we use 25 micromolar zinc, and this gel contains 20 micromolar, so more or less exactly the same. Women that had been suffering from recurrent vaginal infections were asked to use this zinc containing gel, and this is what we saw here in this pilot study. The individuals that it turned out had been suffering from recurrent bacterial vaginosis. Nothing happened. The zinc gel had no effect, and this is unsurprising because bacteria don't have PRA1. I'll come back to the evolutionary origin and fate of PRA1 in a couple of slides' time. But the women that had been suffering from recurrent vaginal candidiasis, so this is like very, very regular reinfections. Once they started using this zinc-containing gel, the vaginitis recurrence didn't reoccur. So the zinc gel appears to prevent reinfection in women with recurrent vaginal candida infections. Okay, so I've shown you that a low zinc environment triggers the expression of the PRA1 gene, and the PRA1 gene produces the PRA1 protein because the fungus is trying to get zinc. But unfortunately, in the context of vaginal infection, that PRA1-induced inflammation is driving disease. And in our in vitro models, ex vivo models, in vivo models, and even in this pilot study in humans, adding some zinc is able to block the whole pathway. Okay, so that was in Candida albicans. Candida albicans is responsible for around 85 to 90% of vaginal candida infections. The second most common cause is a different species called Candida glabrata. Now, whilst Candida albicans has got the PRA1 gene, Candida glabrata does not have the PRA1 gene, right? And if PRA1 is driving inflammation, what's happening here? So we looked into the clinical setting, and what we found, these are vaginal swabs again, number of neutrophils in the vagina of uninfected women, so very low neutrophils, this was known already. Candida albicans infection, very high neutrophil count, this was already known. But in the vagina of Candida glabrata infected women, actually the neutrophil levels were very, very low. So there's a very big difference between Candida albicans infections and Candida glabrata infections, and we postulated that may be due to the presence and absence of PRA1 in these two quite distantly related Candida species. Okay, so we wanted to see if we could recapitulate these clinical observations back in the lab. So what we did was this. We did our ex vivo neutrophil chemotaxis assay again, right? So negative control, no neutrophils come through, positive control, neutrophils come through in high numbers. This is SC5394. This is a well-known Candida albicans clinical isolate. Supernatant of that contains PRA1, drives strong neutrophilic response. These are two Candida glabrata strains. No neutrophils come through in response to either Candida glabrata wild type strains. So actually in our ex vivo infection model reflects the clinical situation quite well. Okay, to see if this may be being driven by PRA1, we took a molecular genetics approach. And what Jane Usher did for us was take PRA1 from Candida albicans and transform it into Candida glabrata with a constitutive promoter. So heterologously express PRA1 in Candida glabrata, which doesn't normally have this gene out in the wild. And this is quite a staggering effect, actually. See here, there's no neutrophils that come towards Candida glabrata at all. We put the PRA1 gene into Candida glabrata and we boost neutrophil recruitment to basically the same level as Candida albicans. So simply by expressing the PRA1 gene in this otherwise unrecognized yeast, turns it into a strong neutrophil chemoattractant. Pretty cool. Okay, so this is the summary of this part of the talk. I'd argue that PRA1 may define the immunopathology of vaginal candida infections. We have PRA1 being expressed and secreted in response to a low zinc environment because the fungus is trying to use it to harvest zinc from its environment. But mammalian immunity, and it's probably happened quite a long time ago because mice and humans are doing the same thing. We get robust IL-1 beta, IL-8 expression, we get lots of neutrophils come in. So innate immunity is targeting PRA1, which in the context of Candida albicans vaginal infections is detrimental to the host because that inflammation is pathological and non-protective. Okay, and because we understand the function of the PRA1 protein, we understand the genetic regulation, the transcriptional regulation of the gene. We have quite a blunt but apparently effective tool for preventing Candida albicans vaginal infections simply by adding some zinc, switching off the PRA1 gene, switching off the PRA1 protein, preventing inflammation, preventing disease. So that's the idea. And then, of course, this seems to work actually in the pilot study. We're doing a larger clinical trial now. But of course, in the future, what we want to do is develop maybe more sophisticated, targeted methods for manipulating this inflammatory pathway during infection. Okay, so vaginal candidiasis. Candida albicans, it's associated with inflammation. It's a detrimental inflammation. The neutrophils don't clear the fungus and seem to drive disease. So inflammation in this case is bad. Hopefully I've persuaded you that PRA1 is at least involved in driving this inflammatory response in the vagina, and quite neatly we can downregulate PRA1 and block inflammation with the addition of a little bit of zinc. Candida glabrata, on the other hand, well, there's no neutrophils, right? There's no PRA1, and I think that's probably the genetic basis for why there's no neutrophils. But the fact that we don't observe neutrophils in the clinic, we don't see a neutrophilic response in our model. And this nice paper from Paul Fidel's group didn't see a neutrophilic inflammatory response against Candida glabrata in vivo vaginitis model, suggests to me that the diseases caused by Candida albicans and caused by Candida glabrata are two distinct types of infection, and they should be considered as such. Okay, so those were two species. Candida albicans, it's got PRA1, it recruits neutrophils. Candida glabrata has lost PRA1, it doesn't recruit neutrophils. What about in other fungi? Because I guess in the audience there will be people working on Cryptococcus, pneumocystis, aspergillosis, etc. So let's have a look at the PRA1 ortholog in some of the other fungal species. Back when we first did our Zinc work on PRA1, we didn't have very many genomes to look at because it was back in 2012. This is all that's all we had. And what we noticed was Candida albicans was present in some species and absent in others. So present in albicans, absent in glabrata, right? But we had a relatively limited number of fungal genomes to look at the time. Now, if anything, there's too much for me to get my head around, at least. There's absolutely tons of fungal genomes out there. And obviously, we can't look at every fungal species on the planet. There's an estimated 3 to 5 million of them, but we can take a variety of approaches and look for the presence or absence of the PRA1 gene in fungi. And what we find in general, it depends on the sample and the location, etc. But in general, around 75% of all fungi, if we look at unbiasedly as fungi as a whole, around 75% have got the PRA1 gene. And it is found in members of the Ascomycota, all those purple guys here, the Basidiomycota, all these red guys here, which make up the dicarya. And what that means is that it emerged in the last common ancestor of the dicarya, which means that actually if you're working on a human fungal pathogen, the PRA1 gene was around in the last common ancestor of all human fungal pathogens on the planet, with the exception of the zygomycete mucor lineage, which diverged a little bit earlier. So it's old, right? It's present in most. What about invasive human fungal pathogens? Right, so this is all of the ones that I could think of. If I've missed any, let me know in the chat and I'll be happy to add them. Right, so you might have noticed from that tree earlier that Candida albicans has got the gene, Candida glabrata has lost it, but also Oris has still got the PRA1 gene, Lusitania has lost it, Guillermondii has got it, Tropicalis has got it, Parapsilosis has lost it, Krusei has lost it, so quite a complex little pattern of gene loss events in the Candida clade. Aspergillus is a bit more normal. Most Aspergilli have got the PRA1 ortholog, including Aspergillus fumigatus, which is called ASPF2. They also have interactions with the immune system, and it's only been lost apparently twice in the ancestor of Niger and this little group here in the Aspergilli. In the Onyginals, which are the endemic dimorphs, blastohisto, cocci, etc., quite a complex picture. Blastomyces, well, up until yesterday, I thought all blastomyces had PRA1 ortholog. There's a new one that's lost it. But in general, blastomyces have got PRA1. Histoplasma have lost PRA1. Amergomyces africanus, the new kid on the block, lost PRA1, while its relatives have kept it. And coccidioides down here kept it. It's quite a complex picture of retention and loss of this inflammatory molecule in the endemic dimorphs. And finally, Cryptococcus, the Cryptococcus, the virulent Cryptococcus species complex, Neoformans gattai, they've both lost the PRA1 gene, probably in the last common ancestor, because it's present in relatively closely related species. So really quite complex loss events in the human fungal pathogens. So if we look at what I'm calling all important invasive human fungal pathogens as a whole, actually around 65% of them have lost the PRA1 gene, and this is in contrast to 75% of fungi in general have it, still having it. So there seems to be an enrichment for PRA1 gene loss in invasive human fungal pathogens. Albicans, cocci, and Aspergillus fumigatus are almost the exception in still having the gene. Okay, so I'm going to show you one slightly speculative piece of data that is addressing a novel hypothesis stemming from this inflammatory pathway. Can PRA1 gene loss protect fungi from neutrophil killing? Okay. So we designed this experiment here. It's a little bit convoluted and a little bit artificial, but this is what we did. We cultured Candida albicans under conditions where the wild type will express PRA1, and then we add some neutrophils to the upper compartment of the chemotaxis plate. We allow them to migrate through for like an hour, then we remove the insert and allow the neutrophils that have got through to do a bit of killing, then we get rid of them, then we assess candida viability. Okay, so it's just to show that in principle, the loss of PRA1 in fungal lineages, in this case by my hand, because I deleted it in Candida albicans, in principle can not exactly protect the fungi from the neutrophils, but you know, if neutrophils don't reach the fungus in the first place, they can't kill, right? So it's just that in principle experiment that loss of PRA1 may have a positive benefit on certain fungi such as Crypto, Histo, Candida glabrata, etc. Okay, so this is the model. Most fungi, 75% of fungi on the planet that we've looked at so far have got PRA1. If they landed in a normally sterile anatomical niche, they're going to be hit with nutritional immunity straight away. They're going to be hit with a low zinc environment. The expression of PRA1 in every fungus we've looked at so far is hardwired to a low zinc environment. So when fungi that have PRA1 see low zinc, they upregulate the gene. They can't do anything about it. The idea is neutrophils will be alerted to this neutrophil chemoattractant, will come in and in most cases clear the fungus. Obviously, Candida albicans has got a few more tricks up its sleeve, and its PRA1 has actually evolved really cool immune interaction properties as demonstrated by Piers Zitfeld's group and others. But the idea in the human fungal pathogens, the Histoplasma, Cryptococcus, etc., by losing the PRA1 gene, of course, there's other inflammatory molecules, beta-glucans, etc. But the idea is that by losing the PRA1 gene, they're avoiding this arm of nutritional, of innate immunity. So the neutrophil attraction that we see against PRA1 is maybe absent. Okay, so I know that's not the be all and end all, but what I'm suggesting is that the PRA1 gene loss events in some of these fungi, at the very least, may influence inflammation during the host pathogen interaction. And my hypothesis, which we're now pursuing, is that PRA1 gene loss events in the ancestors of fungal lineages that are now become pathogens may contribute to immune evasion in terms of not recruiting neutrophils during infection. Okay, that just leaves me with the final model in the last minute. Candida albicans produces PRA1 in response to a low zinc environment, tries to harvest zinc for the fungus so that it can grow, but our innate immune system has evolved to target PRA1, so we have neutrophilic responses towards PRA1, we have upregulation of pro-inflammatory cytokines in response to PRA1. And in the context of vaginal Candida albicans infections, this inflammatory response seems to be detrimental because it fails to clear the fungus, provokes inflammation, provokes symptomatic disease. However, in other host niches, the expression of PRA1 may actually benefit the host by provoking a healthy neutrophilic response and fungal clearance. And that may also be the case outside of Candida albicans. We now have evidence that PRA1 molecules from other fungal species can promote similar inflammatory responses by the host. Okay, that is me. That just leaves me to thank my wonderful Zinky team in Exeter, Alex, what's happened to my mouse here, Alex, Jana, Elena, and Tanmoy. As I said, Elena did most of the work described here, and we hope will be published in the not too distant future. Thanks very much for your attention. Back to you, Gordon.
Duncan, thank you for a wonderful talk. Just to remind everyone that please submit your questions in the Q&A box with the speaker's name, and I will hand back to Joe and we can start up on the questions.
Terrific. Thanks very much, Duncan and Gordon. We'll lead off with a question for Agostinho from Neil Gao. And Neil says, great work, Agostinho. Is it possible that the melanin mutant effect on lactate glycolysis was due to, or even in part, to the exposure of underlying GAG in the swollen spore rather than the melanin directly? Did you check GAG mutants, and did purified melanin by itself affect metabolism?
So we did check GAG mutants. They do not have a significant difference in terms of glycolysis using the model system that we used. but however, I do agree with Neil. So there's something going on that we really do not know what it is. But basically, when we use the albino mutants, the loss of lactate secretion is not complete. So basically, there is also something that is expressed by the albino mutants that is providing a sort of slight activation of this pathway. Now, the other question has to do with melanin. We actually did stimulate macrophages with melanin ghosts. There was no effect. Actually, there was also no effect when we just added in combination melanin ghosts and albino spores. We only saw an effect when we had actually the live spore coated with melanin. Okay, if we used inactivated spores, there was no effect. So to us, this seems to suggest that actually melanin needs to be delivered inside the phagosome, and the fungus being alive starts to germinate. And it's this removal process that somehow activates then mechanisms that we know that lead to mTOR recruitment, but we don't know everything about it. So we need the live fungus and we need melanin to be removed inside the phagosome.
Great. Over to you, Gordon.
Right, Duncan. So questions for you. So let's talk first of all about the clinical picture. So there's a question here about the reduction of the recurrence in the woman. Was it symptoms only or symptoms and culture? In other words, how was it defined that you got a reduction? And then a sort of related question was, does this work on other skin infections, for example, thrush or nappy rash?
Okay, so the first question, that pilot study was done with a small number of individuals, and that was just reported symptoms. We're now doing a proper clinical trial, controlled clinical trial with a larger number of individuals, and in that we'll be able to monitor clinical parameters as well as cytokine levels, PRA1 expression, etc., over time. So should be able to answer that question better in, I don't know, about a year and a half. We just recruited our first participant two weeks ago, actually. The second question, other infections. Good point, right? So Candida albicans skin infections are also seem to be a maladaptive inflammatory response, right? Incidentally, skin is the other human niche, which is acidic, and possibly nappy rash or diaper rash for you Americans. Cream contains a very high concentration of zinc oxide, which is actually way more than is needed to switch off PRA1, but would definitely switch off PRA1 if it didn't kill Candida outright. So yeah, I think that my postulation here, obviously we're interested primarily in Candida albicans vaginal infections here, but if we look more broadly at any fungus and its PRA1 status, and then all potential anatomical niches and potential infection, the absence and presence of PRA1, whether due to gene loss in an evolutionary context, or upregulation or repression by the low zinc or high zinc environment, I think could impact lots of fungal infections at lots of stages, which is something that we're trying to follow up now.
And then in terms of the pH, so there's a question here. So you did these in vitro assays with your neutrophil migration assays, and you showed, I think very early on in your talk, that the pH didn't matter. But did you try those experiments where you used fungi grown at those two different pHs or not? For the 5 and 7, sorry. For the
neutrophil chemotaxis assay.
Yeah. Did you try?
Yeah. So we what I presented there was standard RPMI, which is pH 7, because we do get a stronger in vitro, just with media, we do get a stronger PRA1 upregulation at neutrality. For some reason, the vaginal epithelia seems to further stimulate PRA1 expression beyond that. But for the chemotaxis, where we just wanted the PRA1 produced, we did those at pH 7 because PRA1 is more highly upregulated. But we did do the neutrophil chemoattraction assay in media at pH 5, and also provoked a response, but not quite as strong in vitro when we just did PRA1 and neutrophils and nothing else in isolation. But yeah, it did work.
Okay, thank you. Joe.
Agostinho, a quite interesting finding that mTOR activation and HIF1 are required for the stimulation of glucose metabolism in response to aspergillosis. And the question is with respect to inhibitors of those pathways, for example, rapamycin, are you able to take advantage of that? And are there any clinical correlates for patients taking rapamycin for any of the indications?
We did actually in vitro test mTOR inhibition, and we do see we lose the effect on glucose metabolism. We do not have clinical correlates for rapamycin, but we have for other mTOR inhibitors that are also used in treatment of graft-versus-host disease, for instance. And if we think that these patients are actually particularly susceptible to aspergillus infection, we could speculate that this might be linked, but we don't actually have any evidence that supports this hypothesis.
Great. Room for further clinical studies then.
Yeah.
Over to you, Gordon.
All right, Duncan, two questions around the microbiome, or the microbiome in this case. So does zinc perturb the bacterial microbiome? Could that influence inflammation? And second question that's related is, if you're treating with zinc, right, could that then lead to reduction in Candida albicans and promote replacement with glabrata, for example?
Right, so with the bacterial microbiota of the vagina, I don't know, but there was a nice study Manuel Rafatoulis showed that in the gut of mice anyway, Salmonella typhimurium triggers a calprotectin response, so a zinc restriction, which basically wipes out all of its competitors and actually needs that inflammatory response to kill its competitors in order to overtake and grow. So yes, zinc levels can have massive impact in the bacterial composition of mucosa. With regards replacement, the point here is that the zinc treatment doesn't, this is the thing, it doesn't damage Candida albicans in any way, right? So Candida albicans is there, but when it's hungry for zinc, it switches on PRA1 because it wants to get zinc, and that causes inflammation. If we give it zinc, the Candida doesn't go away. Stay at the same levels. It just switches off the inflammatory response. You're talking about what long-term zinc supplementation might lead to gene loss in Candida albicans. Yeah, I don't know. It's possible, but
also replacement with other pathogenic species, I guess, is the question.
Yeah, but why would they come in if Candida is not moving? And so we're not getting. The thing is, this is the point with the zinc. We're not getting rid of Candida albicans.
Okay. Then can I ask you? There's a question here about PRA1. So you showed at the end there about the sort of retaining of PRA1 in some organisms, but loss predominantly in pathogens. So the question is this a fitness cost? How do these organisms get enough zinc to survive, like in glabrata, for example?
Well, they use transporters, right? So all fungi have got a ZRT2, which we published a couple of years ago. All fungi have got a ZRT2 ortholog in which they can take up zinc from their environment, right? Actually, acidic pH and neutral alkaline pH. It seems quite broad in Candida albicans, at least, and all fungi have got it, at least one copy. Some have got 5, right? PRA1 and Aspergillus and some other species have additionally got PRA1. PRA1 is only useful. So if we do growth assays of wild-type Candida albicans and PRA1 deletion mutant, we only see a growth defect at pH 7.5 in the presence of a zinc chelator, right? We take away the chelator, PRA1 is completely redundant. We acidify the media, PRA1 is completely redundant. So although it's been expressed at acidic pH, it doesn't do anything beneficial for the fungus unless the fungus wants inflammation. So yeah, you can lose. I mean, when I first deleted the PRA1 gene, and that 1998 paper with Bill Fonzi, I think, you know, they saw its expression levels change. They didn't see a phenotype with a mutant, and you know, we have to design our growth assays extremely carefully in order to see phenotypes with the PRA1 deletion mutant, because if we free grow in YPD, it just uses vacuolar zinc to grow. It doesn't need PRA1. If we grow in acidic pH, PRA1 is not essential for zinc scavenging. It's not involved in zinc scavenging. And similar, if we remove a chelator or a competitor. So it's in quite specific environments that PRA1 plays a crucial role, but they must be relatively commonplace for 75% of species to have maintained the gene.
So what about expression of PRA1 during sepsis or during systemic infection? Have you looked?
Not specifically, but I would imagine it would be strongly, very strongly upregulated in systemic infections, at least in the kidney, in the bloodstream for sure, because we've got low zinc, we've got neutral pH, which is optimal PRA1 induction conditions. Similarly, in the kidney is low in zinc. In the liver, maybe not, because during the kind of acute phase response, along with the anemia that makes us go pale and the fever that makes us feel hot, we also suffer from zincemia, which we don't see because zinc is invisible. And the way that is achieved is by shuttling extracellular circulating zinc into the liver. So the liver actually can be quite high in zinc during invasive infections in general. But yeah, in kidney, bloodstream biofilms, I would imagine PRA1 will be strongly upregulated, and it is upregulated by biofilms, as demonstrated by Aaron Mitchell's group.
Okay, thank you. Back to Joe.
Agostinho, there are prominent examples of human genetic variation associated with resistance to infectious diseases, things like sickle cell trait and sickle cell anemia to be protective against malaria. The examples that you've described for natural human variation and metabolic pathways are very striking. And how do you think about those? Do you think that they evolved in response to selective pressure from fungal pathogens or other pathogens? And do they confer any kind of cost similar to sickle cell anemia?
Well, I would say so. I think it's possible. I think there's a couple of examples, not of resistance, but of susceptibility that we've been, classically attributing, for instance, to defects in ROS production, for instance, chronic granulomatous diseases. And we've always thought, it is possible to basically rescue those cells if we somehow would be able to put ROS back in there. So there are some ongoing studies that do seem to show that by manipulating, for instance, cell metabolism, you can rescue the antifungal effector functions of those cells without even touching ROS production. So I would say that, in my opinion, these fundamental processes of cell biology, basically, they do seem to be more important than we realized so far, just in terms of what the cell does and how the cell feeds. I think it's also important for its function. So I think that it is possible that there are a number of variants that are associated with resistance or susceptibility, and for some of those we really don't know exactly the mechanisms. For instance, it could be the case that some of these more fundamental processes are also important in the phenotypes.
Great, thanks very much. Over to you, Gordon.
Well, I have two more questions, one for Agostinho and one for Duncan. So Agostinho, so George just showed that the melanin, the type of melanin didn't matter, right? That in his paper, that DHA melanin and L-dopa melanin both did the same thing. You've only looked at DHA melanin. Does L-dopa melanin do the same thing in terms of metabolism?
Yes, it's the same thing. So in the sense that if we use a L-dopa mutants well, we used Netherlands mutants, we used other systems, and we do see the same thing. Actually, it's interesting. It is not DHN or DOPA melanin specific, because we can even use synthetic melanin. If we just take the albino mutants and we coat them with synthetic melanin, we can also see some rescue. Not as good as when we use aspergillus purified melanin, but we do see some effect. Whether this is related to other characteristics of the pigment, we don't know.
Okay, interesting, thank you. And then Duncan, my last question for you is something about zinc recycling. There's a question here is, Considering that zinc itself is not metabolized, is it recycled?
Yeah, well, yes, and zinc in nature, zinc is only created when a star undergoes a supernova. That's the only way it's made in nature. And I'm not aware of any natural process of maybe the end of the universe which will destroy zinc. So yes, there's it is recycled, and human fungal pathogens get it from their host. We get it from eating, well, beef, shellfish, and
I guess the context here, though, is during the infection. So there's a local concentration of zinc. Is it recycled locally? I guess is what the question is really aiming at.
well, I mean, the host usually attempts to manipulate that that aspect of it by restricting the zinc. So yeah, I mean, in principle, it's mucosa. Mammalian zinc uptake transporters can actually physically reduce the number of zinc ions at the mucosa, and then at the same time, even in the presence of the zinc ions at the mucosa, the secretion or the release of zinc binding proteins such as calprotectin and other S100 proteins can further limit access. But I'm not sure I fully understand in terms of zinc recycling.
Okay.
Because it's there and it can be transported and shuttled. Let's say it is shuttled out of the circulation into the liver, for example, as I said earlier.
Okay. Yeah, I'm trying to just present the question as given. I'm not quite certain in the context it is. So thanks very much anyway. Right, back to you, Joe. I have no more questions.
Well, I think that brings us to the close of MycoTalks. So it just falls to us to thank our two illustrious speakers for being here today and presenting their scintillating science. And thanks to Gordon Brown for inception of this series and its broad impact across the community and the planet.
Thank you. Well, the team behind it, it's Jules and Alberto and the others who run the show. It makes it very easy for us. So thanks to them too. Thanks very much.
Absolutely. Thanks very much. Have a good rest of your day.
Goodbye, everyone.
Thanks, folks. Bye-bye.
Bye-bye.